All right. Today we're putting on our detective hats because we're diving deep into a mystery, a really complex one that's happening right now inside our own cells. We're talking about Parkinson's disease. So, first things first, let's just lay out the stakes here. By 2030, it's projected that a staggering 8.7 million people worldwide will be living with Parkinson's. Now, to put that in perspective, that's more than double the number from just 2005. This isn't just some abstract statistic. This is a global health crisis, and it's growing fast. So, that leads us to the big question, right? The central question of our entire investigation. What on earth is causing this? What's going so wrong inside the brain to cause such a devastating disease? Well, to find out, we have to go right to the cellular scene of the crime. All right, our investigation starts right inside the brain, in a part of it that's, well, it's basically been taken hostage by this disease. Okay, so at its very core, Parkinson's is a story of loss. A tragic loss, really. We're losing a very, very specific kind of brain cell, the one that produced dopamine. Now, these cells, they live in this tiny little area deep in the brain called the substantia nigra. Think of them as the conductors of your body's orchestra, especially when it comes to movement. But in Parkinson's, these absolutely vital cells are just dying off at an alarming rate. And of course, when these neurons die, you start to see the telltale signs of the disease. The tremors, the stiffness, the slow movement. But, and this is the really insidious part of the whole mystery, the most obvious symptoms, the ones you can actually see, they only show up after 50 to 80% of these neurons are already gone. I mean, think about that. The damage is already deep and widespread, long before the alarm ever gets raised. So, as detectives, our first question has to be, why? Why are these specific cells dying? And our first major clue? It points to a massive energy crisis happening at the cellular level. Just imagine your brain's neurons for a second. They're like this huge, bustling network of superhighways, right? They're constantly firing, sending signals, managing literally everything you think and do. And all of that activity, it takes a colossal amount of energy. I mean, these are some of the most energy-hungry cells in your entire body. So, you gotta ask, right? What happens when their power supply fails? If these super high-demand cells can't get the fuel they need, what happens then? Well, the whole system just grinds to a halt. And this is where our investigation points us directly to our prime suspect. And here they are. Meet the mitochondria. You probably remember them from biology class as the power plants of the cell. And that's exactly what they are. These tiny little engines are responsible for generating almost all the energy, the stuff called ATP, that our cells need just to survive. So for those super energy-hungry neurons we were just talking about, healthy mitochondria are, well, they're absolutely non-negotiable. And here it is. This is where the case starts to break wide open. In Parkinson's, the mitochondria are failing. There's a mountain of evidence now that shows this dysfunction is a central, key feature of the disease. The power plants are going offline, and that creates this catastrophic energy shortage right in the very cells that need that energy the most. But here's the thing. A failing power plant doesn't just stop working. It starts to get inefficient, it starts to produce toxic pollution, and in the cellular world, this pollution, this is the weapon. This is what causes immense damage. And this weapon has a name: oxidative stress. The best way to think about it is like cellular rust. See, those faulty mitochondria, they start spewing out these really damaging molecules called reactive oxygen species, or ROS for short. Normally, the cell has defenses for this, but it just gets overwhelmed. And these ROS molecules start to rust and damage pretty much everything they touch. But it's not just a one-way street of damage. It's way more sinister than that. As this quote from the research puts it, we're looking at a vicious cycle. Let's break down exactly what that means, because this is crucial. Okay, so this feedback loop you're seeing, this is the key. It all starts with those faulty mitochondria producing that cellular rust, the ROS. Now, this rust damages all the vital parts of the cell, including a protein that's normally helpful called alpha-synuclein. The damage causes it to misfold and form these toxic clumps. And here's the kicker. These clumps then go and attack the mitochondria, which makes them fail even more and produce even more rust. You see? The cell gets locked in this horrifying downward spiral of destruction it just can't escape. Okay, let's recap our investigation. We have our suspect, the failing mitochondria. We have our weapon, oxidative stress. And we have the victim, the neuron. But where exactly is the final fatal blow delivered? Where does all this lead? Well, the answer is a pretty surprising twist in our case. So that brings us to the microscopic crime scene itself. Where are these toxic protein clumps actually born? It turns out the very birthplace of all this destruction was originally designed to be a safe haven. So inside all of our cells, we have these things called stress granules. Now, under normal temporary stress, like, say, a brief shock, they act like these little emergency shelters. They form really quickly to protect all the important stuff inside the cell, and then once the danger's gone, they just dissolve. All good. But under the kind of chronic, relentless oxidative stress that we see in Parkinson's, these shelters, they transform. They become these persistent pathological prisons. They trap proteins inside, and they become the exact spot, the toxic ground zero, where this horrible aggregation begins. And this diagram just illustrates that transformation perfectly. Look on the left, you've got a normal, protective stress granule, the shelter. But when it gets bombarded by that chronic oxidative stress, like you see in the middle, it changes. It becomes this dense, pathological trap, the prison. This becomes the crime scene. This is the nucleation site, where those toxic clumps of alpha-synuclein are actually formed. And what's really fascinating, or maybe a little terrifying, is that this whole mechanism, it's not unique to Parkinson's. Just look at this table of the usual suspects. This whole sheltered-turned-trap scenario is a key player in other devastating neurodegenerative diseases too. For Alzheimer's, it's a protein called tau. For ALS, it's TDP-43. It really seems like these pathological stress granules are a common thread, a common crime scene, linking many of these conditions. So now that we've mapped out the crime scene, we've identified the culprits, our investigation can finally pivot from just understanding the problem to actually finding a solution. And this new understanding gives us a really clear path forward. And having this level of detail, it's everything. Instead of just taking shots in the dark, researchers can now design therapies that precisely target the key players in this whole destructive cycle. So this insight leads to this really powerful three-pronged attack strategy. Okay, step 1, repair the power plants. Or rather, clean them up. We can boost the cell's natural garbage disposal system, a process called mitophagy, to clear out all those damaged, faulty mitochondria. Step 2, build new ones. We can actually increase something called mitochondrial biogenesis, which is basically telling the cell to build fresh, healthy, new power plants. And finally, step 3, boost the defenses. We can activate a pathway called NRF2. Think of it like a master switch that just cranks up the body's own antioxidant and protective responses. And this quote from the research just sums it up perfectly. The primary goals are crystal clear. The path forward is all about fixing the energy supply. It's about restoring that balance, getting rid of the bad mitochondria, and building more of the good ones. So this leaves us with one final, and I think really hopeful question. Now that the crime scene is mapped and the culprits are identified, could we be on the verge of turning these cellular prisons back into safe havens? All this detective work at the cellular level, it's not just about solving a mystery. It's about drawing a clear map that could, just maybe, lead us to a cure.